second part at the symposium in honor to Professor Sir Tom Blundell with the ceremony of the Academic Council of San Carlos Institute of Physics that will award Professor Sir Tom Blundell with the Medal of Honor and the Honorary Professor's degree of our Institute. On behalf of the Academic Council of San Carlos Institute of Physics, I invite to take their places simultaneously the Director and President of the Academic Council of San Carlos Institute of Physics, Professor Tito José Boragamba, and Professor Sir Tom Blundell. I invite to take his place the Deputy Director of San Carlos Institute of Physics, who proposed a tribute, Professor Richard Charles Garrett. I invite to take his place the representative of the Academic Council of San Carlos Institute of Physics, who also proposed a tribute, Professor Glaucius Oliva. Thank you, gentlemen. Please be seated. To formally open this ceremony, I invite the Director and the President of the Academic Council of San Carlos Institute of Physics, Professor Tito José Boragamba.
Good afternoon again. I am very pleased to open this special session of the Academic Council of the San Carlos Institute of Physics, University of São Paulo, where we will formally acknowledge the fundamental contributions which Professor Sir Tom Blundell has made to our institute over many decades and confer on him the title of Honorary Professor together with the Medal of Honor.
Thank you. Ladies and gentlemen, please stand for the Brazil national anthem. invite to his speech the representative of the Academic Council of São Carlos Institute of Physics, Professor Glaucius Oliva.
Good afternoon once again. It's a great pleasure to be here representing the Academic Council. I used to call it congregation, but it's obviously Academic Council. I think is a good definition for it, of the Institute of Physics of São Carlos. So I'd like to first thank and congratulate our director here, Professor Tito Bonagamba, my lifetime colleague Richard Garrett, Vice Director of the Institute, and Professor Tom Blundell, a mentor, friend, and supervisor. And especially I'd like to thank the presence of all members of our Academic Council here, the congregation, several of them that are breaking their own holidays, and I'm very thankful for you to be here together with many of our collaborators, ex-students and current students, postdocs, friends. So thank you all for coming. I'll try to summarize some of Tom's achievements that led the congregation of the Institute to agree with the proposition for the award of the Medal of Honor to Professor Blundell. Well, Tom was in 1961 the first in his family to go to university, winning an open scholarship to Oxford University. He gained a first-class degree in natural sciences in 1964, moving to research in the Department of Chemical Crystallography, first with Tiny Powell, FRS, for his doctorate of philosophy, and then with Nobel laureate Dorothy Hodgkin, FRS as well, and was a member of Dorothy's team in 1969 that solved the first structure of protein hormone, insulin. After further research on insulin in Oxford, he moved briefly to Sussex in 1973 to form his own research team in biological sciences, solving the structure of another polypeptide hormone, glucagon. He was recruited as Bernal Professor and Head of Department at Berkeley College in 1976, where he stayed for 20 years. And then in 1996, he moved to Cambridge as Sir William Dunn Professor and Head of the Department of Biochemistry, becoming Chair of the School of Biological Sciences between 2003 and 2009. Since 2009, Tom Blundell has maintained an active laboratory as Director of Research and Emeritus Sir William Dunn Professor in the Department of Biochemistry, University of Cambridge. Tom Blundell researches on molecular, structural, and computational biology, with several and seminal discoveries related to growth factors, receptor activator, signal transduction, and DNA double-strand break repair, important in cancer, tuberculosis, and familial diseases. His group has written software packages for computational protein modeling and design, including the widely used modeler and Fuge. Recently, his group has produced computer programs and databases to predict the effects of mutations on small molecule binding relevant to drug resistance. Tom has published close to 600 papers, and including around 40 of those 600 in Nature and Science. I must say that his last science paper was just a few weeks ago. Tom has developed new approaches to structure-guided and fragment-based drug discovery. In 1999, he co-founded Aztecs Therapeutics, an oncology company that moved 10 drugs in clinical trials, and one of them, which has been recently approved to go to market, and it will be marketed by Novartis. This is something that not many people did in their lives. the company Aztecs Therapeutics was sold in 2013 to Otsuka, a Japanese company, for the sum of $886 million US. So academy can produce wealth as well. In the University of Cambridge, he has developed structure-guided fragment-based approaches to drug discovery for difficult targets involving multi-protein systems and protein-protein interactions. Tom was strongly influenced by the multidisciplinary and multinational approach taken by his mentor, the Nobel laureate George Hodgkin, in the 1960s and early 70s, and by the Birkbeck Department of Crystallography in London University, a multidisciplinary research laboratory founded by John Hodgkin's supervisors, John Desmond Bernal, where Tom was appointed to be the Bernal Chair at the age of 34 in 1976. Throughout his fantastic life, he has always built up multinational, multidisciplinary and gender diverse research teams, which always included international visitors and researchers from India, South Africa, Brazil and China, as well as USA, Europe and the Soviet Union. Tom has had a passionate and long-term interest in politics. I must say left-wing politics. He was elected as Labour City Councillor of Oxford in 1970. So, solved insulin structure and being a councillor in Oxford City Council. A nice combination. And became chair of planning of Oxford, allowing him to stop the planned motorway through the center of the city of Oxford, pedestrianize much of this historic center and create a conservation area in North Oxford. He nevertheless was later being a left-wing politician. He was later a member of Conservative Prime Minister Margaret Thatcher's Advisory Council on Science and Technology from 1988 to 1990. He took a leading role in national science development, becoming founding CEO of BBSRC, the Biotechnology and Biological Sciences Research Council, the equivalent of our CNPq here for biological sciences, from 1991 to 1996, and then chair of the same council from 2009 to 2015, chairman of the Royal Commission on Environment from 1998 to 2005, deputy chair of the Institute of Cancer Research, and president of UK Science Council since 2011. Tom is married to Bansinian Lindsay Banda from Zimbabwe, a senior research associate in Cambridge. Tom has 2 children, and I wouldn't dare spelling their first names, so I just keep with the second ones. First names are all African, but then Klesi, which is his first daughter. I met her when she was born in the UK in 1988, and she's a lawyer. And Lisa, who's a medical doctor, both very successful. And one son by a 3 years marriage, Ricky, who is a computer scientist. Tom's interests are in modern jazz, Carnatic music, and Western opera, walking and international travel. And he does a lot of international travel, I must say. Tom has had a long interest in environmental issues, chairing the Royal Commission on the Environment from 1998 to 2005. There, he oversaw the production of key reports, including those on energy and climate change, chemicals and the environment, the organization of national parks in the Irish and North Seas to prevent overfishing, and many others. Tom Blundell's work has been wisely recognized by election to academies, including election in 1984 as Fellow of the Royal Society, election as member of the European Molecular Biology Organization (EMBO) in 1985, member of Academia Europea in 1993, founding member of the Academy of Medical Sciences in 1999. International recognition has come in his election as foreign member of the Indian National Science Academy, the Chilean Academy, the World Academy of Sciences and from last year he was elected as a foreign member of the Brazilian Academy of Sciences. Tom has received numerous prizes and awards, including the Alcon Award for Vision Research in 1986, Gold Medal for the Institute of Biotechnology in 1987, Krebs Medal, SIBA Medal, Feldberg Prize, Gold Medal of the Society for Chemical Industry, first recipient of the Pfizer European Prize for Innovation in 1998, and Bernal Medal, Royal Society in 1998. In the coming meeting of the International Union of Crystallography, which will be held in Hyderabad, India next month, Tom will receive the EVO Prize, the highest recognition of the crystallographic community worldwide. Tom has been president of UK Biosciences Federation, president of the Biochemical Society, the equivalent of our SBBQ in Brazil, and president of UK Science Council since 2011. Tom's contributions have been recognized by a knighthood in 1997 when he became Sir Tom Blundell. Tom has honorary doctorate degrees from 16 different universities worldwide. Now, why Tom is so important for structural biology in Brazil and for IFSC in particular, our institute. In the late 70s, as I mentioned before, small molecule crystallography was starting to flourish in Brazil when Tom Blundell hosted in his laboratory the leading Brazilian crystallographer, Professor Mascarenhas, then going for a sabbatical in micromolecular structures. This very successful stay triggered several subsequent visits of Professor Blundell to Brazil when myself, together with other young undergraduate students, were marveled by his fascinating lectures, the fascinating lectures he gave on protein structures and their fundamental implications in understanding life and health. I joined Tom's lab in 1984 as the first of many Brazilian students that he has supported and supervised, directly and profoundly impacting the establishment of protein crystallography and structure-based drug discovery in Brazil. When I returned to Brazil together with Dr. Richard Garrett, who was also a PhD student at Tom's lab, we received the support, encouragement, and guidance in the selection of projects and strategies to establish the first Center for Micromolecular Structures at the São Carlos Institute of Physics and to lead the creation of the first protein crystallography beamline at the Brazilian National Laboratory of Synchron Light, LNLS. Other of Tom's Brazilian students went to different universities and research institutes at various parts of Brazil to create what is nowadays a diverse and flourishing community of research groups in protein crystallography, structural biology, and drug discovery. His generosity and commitment to support crystallographers from Brazil was also effective to scientists from other developing countries, and his lab was always characterized by the broad diversity of nationalities and gender, forging in all of his students a strong view that science should have no borders or prejudices of any sort. For those who, like me, had the honor and privilege of sharing his time, guidance, and friendship, his nomination for the IFSC Medal of Honor is fully deserved to recognize his lifelong dedication to science in general and crystallography in particular. Thank you very much.
At this moment, I invite the Deputy Director, Professor Richard Charles Garrett, to grant the honorary professor's degree of the Institute of Physics of São Carlos to Professor Sir Tom Blundell.
Tom, they've given me the most agreeable of all tasks, and that is to present you with this diploma which formally makes you an honorary professor at the São Carlos Institute of Physics. As you can see, it's Brazilian size, so it's probably twice what you normally expected to get. Everything in Brazil is big, but perhaps what I should do is read it, what's written on the diploma, so that people know what you're formally receiving. I'm sure you'll get the gist of it, and if not, we can explain it later to you. A Igreja e a Congregação do Instituto de Física de São Carlos, na Universidade de São Paulo, tendo em vista a portaria IFSC número 47/2008 e a decisão proferida em sessão realizada em 5 de maio de 2017, confere a Thomas Leon Blundell a medalha de honra do Instituto de Física de São Carlos, com a qual o reconhece como professor honorário do IFSC, por sua contribuição extraordinária ao campo de biofísica estrutural, na compreensão de estruturas de proteínas, sua relação com a função biológica e aplicações na descoberta de novos fármacos, que impactaram de forma determinante esta área de pesquisa no IFSC.
In the same spirit, I invite Professor Glaucius Oliva to grant the Medal of Honor at the St. Carlos Institute of Physics to Professor Sir Tom Blundell.
Bom, I don't have much to say more than what I've just said, but of course you've seen that it's a great honor for us to have you as a member, now an honorary member of our institute, and I want them to give you this medal in this respect.
Ladies and gentlemen, I invite for his speech Professor Sir Tom Blundell.
Well, this is a little bit of a surprise, as I thought I was giving a lecture, but I'd like to say a few words now. So first, thank you very much, everyone, for your very, very kind words, and particularly to Glaucius for his very, very long, almost exhausting, I felt tired at the end of this talk on what I've done. I'd just like to say a few words. I've spent my life, and I'm going to tell you a little bit about it later, traveling the world, because I just love science because it can be so international. It can bring us all together. It has no borders, as Glaucius has said. And I've had a special affection for Brazil because it represents something very important to me, and that is a country that lives with very many different peoples. In my political life, I started by becoming the chair, when I was an undergraduate, of a committee called the Joint Action Committee Against Racial Intolerance. And then I went on to form in the city of Oxford the Oxford Committee for Racial Integration. And Brazil has always been a model for me of how to live together in a very beautiful way. Of course, my wife comes from Southern Africa, from Zimbabwe. We spent a lot of time in South Africa while it was under apartheid, trying to get arrested some of the time, I have to say, but others being as influential as I could be to get South Africa to move towards the Brazilian model. Now, I do realize that I've never quite achieved being properly Brazilian. I'm going to embarrass Glaucius a little bit because I came with my wife and my 2 young children about 20 years ago. The children were very young, I think 5 and something like that. And we were going to the Rio Carnival. And I should say, if any of you have young children, don't just say to them, I'll take you somewhere when you get older. Just take them wherever you go. And I took my children around China, they lived in India, and one of the things I wanted to do, having been to Iguazu and up the Amazon, was to go to the Rio Carnival. So I said to Gracias, I said, look, my wife's black, I'm sort of white, but not exactly. My children are sort of intermediate. As I was going to the Rio Carnival, I put on an old shirt and shorts, and I had sandals on, and I said, Gracias, I look just like a Brazilian. I feel at home. And he looked at me, he said, Tom, you don't look Brazilian. I said, well, Gracias, what's the problem? He paused, and then he said, you've got socks on. So, you see, I've been on a steep learning curve. But it's a great honor to be here and to have visited so many times and to have so many friends and to have had so many words said to me which have been kind, to be given this wonderful honor here, and also having not worn a tie, I see you provided me with an alternative, which is very splendid. So I have to thank you all very, very much. And I have a few more words to say. I hope you've got a drink afterwards. Yes. Thank you very much.
Ladies and gentlemen, I invite for the speech the director of São Carlos Institute of Physics, Professor Tito José Bonagapa.
Dear Honorary Professor of the São Carlos Institute of Physics, University of São Paulo, Sir Tom Blundell, thank you very much for your important contributions for our institute. I would like to say that they were clearly very, very fundamental for the development of crystallography and structural biology, not only in our institute, but all over our country. And this is fundamental, and we would like to thank you very much for all the contributions we have seen from our colleagues that were talking about the collaborations they have with you. So, thank you very much. Now, I would like to invite the new professor of the São Carlos Institute of Physics, our colleague, Tom, okay, to give your special plenary lecture. I would like to invite my colleagues to follow me. Take your...
Do you have a pointer? Well, I have to thank everyone here, I think, for coming, and I'm very, very grateful for the invitation to come to this splendid afternoon, and thank all of those who made so many kind comments about what I've done. It's really very nice. I only wish my wife was with me and the children as well, as they've been here so many times. But thank you all for just a wonderful afternoon. I'm afraid you've got me for a little bit longer, and I'm going to tell you a little bit about the way I've seen my research and my life in science. And as you can see, although I'm going to talk about structural biology, bioinformatics and drug discovery, I'm really going to talk about science as an international activity and the multidisciplinarity about it as well. It's a wonderful way of interacting with people all over the world and doing things as part of a team. And what I particularly enjoyed about science is the way that we can work together in groups for a common objective. And I'm very pleased that those of you who were at Birkbeck earlier on and more recently in Cambridge have made very kind comments about the interactive nature of our research teams. So I'm going to talk a little bit about various aspects of what I've done, but of course the major developments over recent years have been the human genome, but the human genome and of course that of infectious disease agents like TB is only part of the story. We have to move from understanding the genome to really understanding its function or the function of the gene products. And I think that's almost as challenging as determining the genome sequences in the first place. So we need to know about the very complex systems that control regulation, which might be targets if we want to make drugs. We have to remember that nature increases complexity in order to get selectivity in cell regulation and signaling. And so what we've done as structural biologists so far is to look at rather small systems and try and understand them. But that reductionist approach has to be broadened out. This particular enzyme, and this is the paper we published in Nature originally a few years ago, but just this February, the complete chain tracing of this molecule. This has 4,128 amino acids in it. It's the largest single chain protein with a known three-dimensional structure. But it's a part of a multi-component system, and I'll tell you a little bit about that. So we need to go from the genome to the proteome, and in order to do that, we have to also think not only about experimental methods, but computational approaches as well. And what we've been trying to do is to take the genomes and the strain variation that we find, and we sometimes get given literally several thousand sequences of an infectious agent and trying to interpret them. But experimentally, and if we take this database I'll tell you about, we have only 10% of the gene products defined experimentally. And if we're going to do a phenotypic screen in drug discovery, we need to have some basis for understanding the broader set of gene products. So we need to classify and organize the information, and that means good software. We have to model not only the proteins, but the protein ligand interactions, and we need databases of that. So I spent a lot of time recently trying to extend our knowledge to the whole of the proteome, at least in a simple way. We have a lot more to do in that sense. And then we need new methods of drug discovery. Over the years, we had fantastic developments with huge screening libraries, but if you look at the number of new drugs approved, it really hasn't increased very much. In fact, there was a period when it was seeming to decrease. And so one of the things that I've been doing is to think about new methods of making medicines, and as Klaus has mentioned, using methods of fragment-based discovery, and you heard from Marcia quite a lot about it. We now have our first breast cancer drug on the market as a combination therapy. And you will realize that that takes an average of 14 years, so it's quite a long time to work. So I'll tell you about how we got there, and then I want to just say a few words about the big challenge now, is we may have drugs, but we have resistance. We need to understand that resistance. We need to think about how we can avoid resistance to drugs, and I'll say a little bit about that, and how we can understand it. And that brings in not just biology, cell biology, but it brings in computer and new methods, and this is a machine learning method, and that originated, I should say, in Brazil from our colleague Douglas Pires, who unfortunately can't be here today. So that's the picture I want to paint, but I want to talk about it in a rather personal way. And just to say once more that I'm really delighted to be here at the Institute that I think I visited first almost 40 years ago. I think 38 years ago I was here first time. And wonderful times in Brazil, as I said earlier. Some of the most beautiful sights of the Rio Negro up past. I mean, it's just wonderful on Iguazu. I really love it. But I'm going to start with my main influences. And as Klaus said, I was the first person to go to university in my family. I was actually the first person to stay at school beyond the age of 15. So my parents both left school at 14, 15. We had nobody really with an academic background. But what we had in my family was a terrific music and artistic tradition. And so I spent my weekends with my grandfather painting, and there were musical instruments all over the place. So when I got my grammar school place, I went to a school which was, it was founded in 1600, and this is the buildings where I had lessons in. They were very old, but it was a quite exciting experience. I should say it was a selective system, and I passed it, my sister passed it, no one else in the family had, and my brother failed it. And the successful one is my brother of the three of us, because he's now up on Thomson Reuters to get the Nobel Prize in mathematical economics. So you can see selective systems when you're young are not always a good idea, unless you have a very encouraging family for this sort of thing. But what I did there was really to get interested in music. I had no lessons at all. I just had dozens of instruments, every possible kind, around the house, and so I picked them up and taught myself. And of course, when I was 14, I formed my first jazz group. It was a traditional jazz. And then I got my scholarships to Oxford. By that time, I was in mainstream jazz. I thought I was going to do really art subjects, but I realized that I was probably better at maths and physics and chemistry, so I got a scholarship for that. But the first thing I did when I got there was to find colleagues who were playing music, and we followed people like Charlie Mingus, and I became a semi-professional jazz musician, which lasted quite a time, and I'll tell you how that came to at least a practicing end, although I go to jazz festivals still very regularly. But the other influence I had was in this man, J.D. Blundell, and you've already heard a lot about him, because he was the supervisor of Dorothy Hodgkin and her influence in many ways. But I didn't know anything about Dorothy Hodgkin when I read J.D. Blundell. I read J.D. Blundell because he gave me an idea in his social function of science which was published in 1938, of the importance of science in policy and in development, and I was already becoming rather radical in my politics. I moved slightly right-winged to the right to join the Labour Party, I should say. But before that, I had really left-wing politics. But much of it was influenced by J.D. Blundell, and also reading his Science and History and realising how important it was to understand the social context of science, and also to have science influence the social context. So that was my big influence. And when I decided first that I might do a degree in economics, but then probably not, because I heard about Dorothy Hodgkin's laboratory and Tiny Powell in crystallography, and I can remember running along South Parks Road, which is the main road in Oxford, and dashing into the laboratory and saying, somebody allow me to do research here. I worked first with Tiny Powell, learning how to write computer programs, learning how to do structures, and that was a pretty good training. I then transferred over to work with Dorothy Hodgkin. And the attraction there, you can see this was the team that actually solved the structure of insulin in the end. And it has New Zealanders, Australian, an English lady, an Indian, another New Zealander. But you see that there were three women involved and 3 men, and this was a very unusual structure. And of course, Dorothy was, I think, the second person to get in the Royal Society as a woman. She was also, of course, one of the few scientists, Nobel Prize winners. And the day that I walked into the laboratory, she was on television in Africa, having just got the Nobel Prize and being interviewed. Why was she in Africa? It was because her husband was interested in African politics, and I also, I have to admit, read his books before I knew very much about Dorothy Hodgkin. So it was a very strange coming together. But it had all the features that I really liked. We had lots of parties. You can see how we're all lounging around here. This is actually Thomas Hodgkin. So I had continuous discussions with Thomas Hodgkin. I asked him, for example, how did you get on when Margaret Thatcher was a student of Dorothy Hodgkin's? And that was about 12 or more years before I was in Oxford. And Thomas looked at me. He said, it was the most wasted year of my life. So we had lots of discussions and no impact at all. And of course, we started to travel with all of these things. This is with Dorothy in 1969, when we first had the low resolution structure of insulin. It's not a very good picture. When I went to the meeting of the International Union in 1969, we just solved it. We had the low resolution structure and everything there. But as I walked in with Dorothy to the international meeting, the organizer of it said to Dorothy, you know, we couldn't get a moon rock behind security, so we don't have a plenary speaker tomorrow to start the meeting. And so they were going to have this big thing about getting rock from the moon and doing X-ray studies of it and so on. And he said to Dorothy, would you give the lectures? I heard you got insulin. And I thought that was a fantastic idea, but Dorothy said, yes, of course, I'll say a few words and Tom can give the talk. So I spent all night up with some slides I had of the low resolution stuff. We hadn't made any slides of the high resolution, so I was busy making those. They were on big lantern slides, and I had some overlays, and I had 3 projectors in the end, and I put on one of them the picture of the insulin structure, another one, the electron density, and I wrote the music that Dorothy was humming when she first saw the map. And Dorothy was amazed. She didn't know she hummed, and she didn't know what she was humming. It was very, very interesting. But then it got very exciting in that summer just before the meeting. So we got these models. This is Dorothy looking at the electron density with an interpretation. You see, it was rather old technology in those days. And this is Guy Dodson, who was really the longer-term person working on the insulin with Dorothy, leading to the successful modelling. And this, I just want to make a point, is Dorothy in the 1930s. She started in 1934 on insulin. Why did she start? Was it because it was to do with diabetes? I think probably not. It was due to the fact that companies had purified and crystallized insulin, and the insulin crystals and material were available. And that interaction between academia and industry had a huge impact on me as a young scientist. These are the crystals that are used to treat diabetics, and this was our structure. And you can see how beautifully the hexamers of insulin are reflected in the rhombohedral structure of the crystals. But it really had a fantastic transformation on my life, because I had to go to Wellcome, I had to go to Novo in Denmark, Eli Lilly in the US, and I suddenly found I had a network of people in industry. And people in Oxford used to ask me at my table, because I got a fellowship and used to sit with all these important people, and they say, well, why does Dorothy keep going to companies? And, you know, she's left-wing. And I would say, well, there's three reasons she went to companies. And first is a lot of her best students were there. Secondly, the science is really fantastic. And thirdly, they provide the insulin and much of the technology that we've used to solve this structure and get these papers in Nature. And that was a surprise to Oxford academics. And I just want to show you one other picture, which is my favourite one here. So this is Dorothy when she started the insulin project. This is Dorothy a little bit later than when she solved the insulin structure a few years later. I think it's impressive to spend that time on a single project. But of course, as I often remark, I like to show this because you can see how beautiful I was in those days. So that was my start, and I was in Oxford. So there was a proposal in Oxford to put a motorway through the centre of the town, as Klaus has mentioned. And I was already involved in the city. I formed this race organisation. I have friends all over in the industry, in the town, and the Labour Party asked me if I would stand for them in the election. It was the end of the Wilson period, and Labour wasn't going to win. And in fact, I was the only Labour candidate who won that year and got a seat in the council. And then I thought, well, we don't want a motorway which was going to go right through the middle of the town like this and across Christchurch Meadow, which is a beautiful thing. We need to have a plan. We need to have an environmental plan. We need to have street communities. We have to have street committees. And we'd already been involved in that. And so I got heavily involved. And the next year, we won all the inside parts of the city. And then the following year, we took control. And I found myself in charge of the City of Oxford's planning. And of course, I wanted to pedestrianise this part of the centre of Oxford. And so I passed through the council, stopping all cars to go in many of the areas here. And then I got called down because there was then a Tory government in the centre of in the country, and they called me down and they said, Blundell, you may be in charge of planning in Oxford, but I'm in charge, this is the Minister of Transport, who was called Keith Speed, I'm in charge of planning nationally. And the High Street in Oxford is part of the national route. So you've got to still have a way across Borden Bridge, which is over the side here somewhere. And so I had to change the plan a little bit, but I pedestrianised the centre, I made North Oxford a conservation area, I stopped the motorway for good, nobody's ever tried to change that. It's one of the few things that I've done which has stayed, and you see how important this is. North Oxford, beautiful old houses, and this is the centre, and we protected that from stupid traffic measures. So I think we were the first ones to have a pedestrianised centre of the city and get rid of cars, and I think that has really influenced my view about the environment, and chaired the Royal Commission on the Environment for many years, and so on. But this is what happened. I was really getting later papers in the morning and early afternoon, which is fantastic. I was in the council offices in the late afternoon, and I was running my modern jazz group in the night, and I didn't get much sleep, and life got rather chaotic, and family life became challenging, and I just got up and I left Oxford, and I took the train to Moscow, and then I wandered across Siberia. I tried to get into China down here, but I couldn't get in because there's a Sino-Soviet war, and I was just on my own. I got stuck on the border around here in Khabarovsk, having gone across by train. Eventually I found a little plane and a beautiful Japanese lady, and I managed to get across to Japan. And then I wandered down from Japan. I started to learn Japanese. My Japanese is not too bad, and another company's been sold to Japanese. I've been using it to introduce and give talks in Japanese. But then I moved down to Taiwan, where I knew there was an opera singer who was a friend of mine, and then down to Hong Kong, where I had another Chinese opera singer and got involved in that. So I got heavily involved in Chinese operas, and even over the last years I put on funded Chinese operas in London. But then I moved on further to India, to Bangalore, and then I found a new passion, and that was Indian music. And I found a lot of similarity between Indian Carnatic music and the Veena. So I took lessons in the Veena. This is actually a friend of mine who's still a semi-professional, well, he's still a professional Carnatic music player, and I get him to play in concerts quite often when I'm involved in meetings in India. So that's all great fun, and I was enjoying myself tremendously. And then I wondered what I was going to do with my life. I mean, music was really rather difficult. I wasn't that good at it. Politics was impossibly difficult. I thought I had to go back to science. So after I went back home, I went to see Dorothy Hodgkin, and she said, Tom, I think you better go somewhere else. And so I was sent off to Sussex University, where I knew quite a lot of people, and Dorothy fixed me up with a lectureship. And so I started working on the functionality of insulin briefly, but then this is the yin and yang of blood sugar control. This puts it down, this puts it up. And I began to think about how receptor interactions work and how these molecules are functioning. It was my first sort of entry into cell regulation and signaling, which became my passion. But then I got distracted further. I started looking at a pepsin, which has been a molecule that Blundell and Hodgkin did a first experiments on, but it had never been sold. And I started then working on relatives in yeast to get the structure. And then I realized that they were related to renin, which controls blood sugar. And so I made a model of it in the late 70s and proposed that we could make drugs by looking at this cleft and also keeping hydrogen bonds but changing all the chemistry. And so the model was we kept the hydrogen bonding but we filled the pockets in a different way. And this I didn't have courage to publish because it was all modeling, but eventually I submitted it to Nature. I think it was about 5 years after we'd built the models of renin, and it was published there. But this became one of the first structure-guided drug discovery efforts. And then another amazing thing happened, and that was Brazil. And this is, you see this picture many times, but you see this is Ivone. I'm very pleased that she sent me a message, and she's been a long-time inspiration and connection. And she came with Sergio to England and spent quite a few months and published papers. And then, as she went away, she said, "I'll send you somebody really good for your lab." And of course, that really good person was Gracias, who turned up a little bit later. And I think I came to Brazil in between, just after that. So that was my entrance to Brazil. But I still kept traveling, although I came back. And this time I found I could get back into China, which I hadn't been able to in '72, because they'd also worked on insulin. And this man here is Liang Tung Chai, who'd worked with us in the 60s, and I knew him very well. And he'd set up and worked during the Cultural Revolution on insulin. And this is me, as you can see, but this is Dorothy. We went to China. This is the main professor in the University of Peking, and somewhere here is Tung Chai, the mark here. So I had to give lectures. So I gave lectures not in Chinese, although I could read some of the characters. I couldn't do the Chinese, and learned the characters through Japan, but this lady translated for me, and I stood below a picture of Mao, and of course everybody was wearing Mao suits. But I found out afterwards that I was required for other things as well, and you can see not many people look as happy as these people do when I play musical instruments. But you can see they're all sitting in their Mao suits, or a lot of them actually, but some of them. But in those days you had to wear these suits, and if it got cold in Beijing, then you put a jersey underneath it. And so my friends who went there couldn't move their arms in the end. It got so cold in Beijing. So it became very international and very beautiful. But going back to the science, about that time, I proposed that this molecule of pepsin and renin had an ancestor that looked like this. I was very heavily criticized by other crystallographers because I was speculating too much. But nevertheless, I published this paper in Nature predicting that this existed. And then every Saturday went and looked in the library to try and find it. And I found it in Rous sarcoma virus. And then I realized it was in HIV. And then I knew that this was a relative of that. So if I had molecules that I could inhibit this with, would they work in that? So just as HIV broke out, and we had the sequence of HIV, and I realized just here was the protease. Other people commented on it, but they didn't understand what the structure was. And I was immediately able to transfer everything I'd done against blood pressure for renin. And I remember going to Pfizer, as I was doing regularly, every other month in US and then in UK, and saying, I want to take the blood pressure lowering molecules we've got and try them in HIV. They thought I was crazy. They said, but one's blood pressure, the other's virus. I said, yeah, but it's going to work. Just try it. And so we took them over. And a lot of people, I think, had similar ideas. But immediately we could show that HIV protease was inhibited. So what we had was the protease as a pair of scissors cutting up the proteome, the protein, the polyprotein that's made. And I didn't publish my models of it, which I had, although my colleagues in the lab did, but eventually we got the structure of this, and this became the basis to make the first HIV inhibitors. So this we published in, as you see, 89. My friend Alex Blundell independently had a structure. Merck did a structure and got it slightly wrong. So we corrected that. and this became the basis. And within 6 years, the companies using these structures had HIV inhibitors on the market and used in AIDS patients. So this became a complete paradigm for me, but I wasn't able to continue this because, to my surprise, I got recruited by Margaret Thatcher to advise her, as you heard, and then I was asked to rearrange the research councils and become the head of those, and I discovered that I could neither do politics nor I could do industry, and so at that point I knew I couldn't be a Marxist entrepreneur for a little while, and so I had to stop working on the pharmaceutical side and designing these things, but other people took the structures through, and we got 4 different HIV drugs onto the market in the next few years. And so this was my paradigm. So when I finished my 6 years reorganizing the research councils and I became able to be involved again in pharmaceutical drug discovery, I realized that if you could go from the gene to the structure, and we've done that by experiment and modeling, that's sort of exploring biological space. And if you went from the structure to the drug, that's exploring chemical space. So what I decided to do in '96, when I came out, was to set up, apart from my academic lab, and the Cancer Research Campaign that funded me all through my times in the government, is to set up that sort of thing. Now, that all went on, and I should mention, while these people were around, I'm borrowing lots of slides here, so you've seen this before, but Glacius had arrived, and Richard was here, and they were all sitting in the same room, and as you've heard, they came back here, both of them, and had a very big impact. And one of the great pleasures, of course, has been seeing what's happened from that period of time in the 80s, and as you heard, Glacius was working on several different projects, but this was all going on in the lab in parallel, and it's great to see what you've done here since you've been back. I feel it's just fantastic. So going back to this, how do we look at biological space? So I'm going to speed up now, otherwise you won't get a drink. And that is, so it became apparent from my other work, and I did structures of many, many different growth factors and hormones, nerve growth factor, FGF, MET, and HGF, and so on. They all involved in very complex systems. This is the simplification of DNA double-strand break repair. There are actually about 12 components. And so what I've begun to do to define biological space is to look at all the components independently. You heard about one, I'll show you a bit more of DNA PK. And then look at them in binary interactions, and then try to define them as more complex systems. So that's the way we've been going. And so we use small-angle X-ray scattering. We do a lot of computing. So one of our 3 papers in Science over the last 3 years or so has been a model of one of the components of this system. And we spotted that through computational analysis of the human genome. A physicist, Takashi Ochi, modelled that. He expressed it in the biochemistry lab. A born and bred physicist, never been in the lab much before, but he expressed the protein. He solved the structure in 2 weeks. He went along to see our colleagues and did the cell biology and ended up with the first author science paper. Not bad. And then we also were doing X-ray, and this is the paper we had in February this year on the DNA PK. And of course, what's revolutionizing everything, and you have to do something about it in Brazil, is cryo-EM. So our new cryo is just going in today. We've been a bit behind the laboratory molecular biology, but I spent a lot of time in the last year with colleagues doing cryo-EM studies. And then you need things like mass spectrometry, nanospray mass spectrometry, to get stoichiometries. So all of these things give you Science and Nature and molecular cell papers. So they're all good science, but they're putting together a picture of a very complicated system. The crystallography is quite challenging. This is an example where we took the selenomethionine version in HeLa cells that nobody else had done before, as far as I know. 120 selenomethionines in each of 2 molecules in the asymmetric unit, so 8,000 amino acids. And superpose those, you can see the green, so we knew we had it. We use those then to help the phasing and also to get the registration of the amino acids in the density. Here's the cryo-EM just compared with the X-ray. You can see they're in pretty good agreement, although this is still only around 7.8 angstrom resolution, maybe slightly better now. And that's the superposition. And then the sort of things we are able to do then is to look and see how these 2 DNA PK molecules organize the DNA so the 2 ends come together. And then you get an assembly on that of all the other components. I'm just going to whip through this, but so what we then do is to look at some of the other components, so XLF and XLCC4, and we can show that these dance together and then form the line dance, then get this incredible structure builds up. And we call this a scaffold, and this assembles to do the repair. You put the components inside the scaffold, and so you've got the DNA PK at the ends of the DNA, but with a scaffold supporting the ligase and all the other components. So you have a stage which is the DNA PK, and you have a scaffold, and you also have strings. So this is my idea about how you organize in space these very complex systems. And I haven't time to show you today, but I also am doing single molecule studies with an optical or magnetic tweezers. We're using magnetic tweezers, and we're looking at these assemblies in single molecule level. So fantastic challenges defining biological space. We put it all in databases I'm going to whip through now, all sophisticated, representing contacts in molecules as structure, interaction fingerprints, new shape descriptors we published in the last few years. Then we link them all into approved drugs. So everything in the protein data bank we fragmented and related to things in approved drugs, so we can immediately pull out any approved drug which has anything similar to anything we find in a structure. And these are all useful tools. It's the importance of doing software as well as experiment, and we do both. And then what we've been doing is taking the human genome and the infectious disease genomes like TB and beginning to model them. And so this is our databases. This is a whole lot of software which some in the public domain, but Jakarta and on the double mapping, and then these programs, Fugue and Modeler, all written in my group, and we use them to construct the proteome. So these are all software. You can see how most of my team have obeyed my request. It was only a gentle request to name their programs after musical terms. So we got composer, we got fugue, and you can see this was written by young people. But I had one difficult guy called Andre Chalet, who wrote this program called Modeler in a year. I said, you know, let's do something similar to what we have in our software to refine proteins, which we do by restraining and putting information. He went away and turned it all upside down, and we ended up with a restraint-based system, so you're satisfying spatial restraints. And this program, as you can see, has been cited 9,500 times. So just imagine being a PhD student for just 1 year and getting a paper with 9,500 citations. That's more than many professors in Cambridge have on all their papers. And and fugue is another one written by another very bright student. So what we do then is put them together in databases. We thought that this is tuberculosis. We thought that Chopin had died of tuberculosis. It may actually be a Mycobacterium abscessus infection, but Bernardo called his software, which shows you can model about 70% of the proteome or the genome, gene products, let's say, pretty accurately. We've got rough models for almost everything, but it means that if you do a phenotypic screen now, you can identify the target, you immediately have models to base your drug discovery and elaboration on. So this is all important. We then take the models, we build models of proteiners, we build models of complexes using lots of docking and virtual screening, and then we use the software that we've developed for modeling all the mutations. And the mutations have become very important, and we have two programs. One is SDM, the other is MCSM. So SDM we published 20 years ago, and it's a statistical method. I'm not going to really explain how you do it, but you look at the probability of a mutation in the folded and unfolded state and set up a free energy system, and you get this formula out of that. I was very proud of this, and when Douglas Pierce walked in the lab, I said, what we need to do, Douglas, is to extend it to the protein-protein interactions and the protein ligand. And Douglas looked at my code. So Douglas is your Brazilian colleague, computer scientist. He looked at my code and he said, God, Tom, this is awful. And I said, Douglas, you know, it's not awful. I've got good computer scientists. What do you really want to do? So what he wanted to do was to use his machine learning methods. And so I said, okay, use your machine learning methods on this problem. And that set off an amazing series of events. So I first thought that Douglas's picture of the protein as a lot of simple vectors wouldn't really be satisfactory. And so I let him get on with it. He's defined the protein not in terms of all the information we had, but a lot of simple vectors between different atoms. And then the residue is the wild type and the mutant be defined as a vector also. And it just seemed too simple. I thought, well, let's see what happens anyway. And of course, like most things when you're running a research team, if you really let people do what they want to do, they come up with amazing results. And to my total amazement, it worked brilliantly. And the reason why it works brilliantly is that my statistical method just takes a local environment, whereas a lot of the influence is about depth into the hydrophobic core, the electrostatics, and I wasn't including that. So now we put the two methods together to some extent. But the method that Douglas has developed, and also with David Asher, so he's in the next slide, this is David Asher. It has been really useful for looking at genetic disease and all sorts of things. But it's just a good example. Is you get somebody who's a computer scientist in the lab and you let them free, but you put them together with somebody who's a biologist and knows about the crystallography, and it works brilliantly. So actually, when we've done some predictions, David took poor Douglas into the biochemistry lab and in a month expressed 30 of the mutations and tested Douglas's thing. Douglas, as a computer scientist, had to do the experiments with David. I think it was really brilliant. But we published, I think, about 20 papers on this, looking at genetic diseases and showing that many of the mutations in these things like this, clear renal carcinoma, are in mutations in interfaces. So they're quite complex and you need structure to understand them. And you can see we can make a drug to combat this by stabilizing the interface, not by disrupting it. So all sorts of ideas come out of it. This is black bone disease, and nobody had looked at it. We found again that lots of the mutations in this disease are also in the interfaces between the subunits. And of course, genetic diseases where people live to 20 or 30 years old, they can't be hugely damaging, otherwise they wouldn't have lived that long. So there's very subtle mechanisms quite often, and we've been finding them. And what's even more exciting is the clinical collaborators that we've had. We've got them to classify the clinical trials according to our definition of the mutations in the disease. So we've run clinical trials on that basis. So you do useful things in this way. So that's looking at biological space. Just very quickly now to go through chemical space. And the idea here, as you've heard from Marcio that we had, was that these huge libraries cover only a tiny bit of chemical space. You need probably 10 to the 40th, or some people say 10 to the 60th molecules of drug-like features to cover chemical space at a drug size. But if you have a small molecule, you only need a tiny fraction of that, because it's a sort of power thing. And if you've got the tiny fraction of a fragment, and you know where it is, you can elaborate it. So what happened is, when I came out of my government job, I first had a proposal for a company to do proteomics, but then I realized I got offered $6 million to start it, and I realized it was a bad idea. I gave it back, and then I came back with a student who was a contemporary of Richard and Glass's, called Haran Jyoti, and we developed the idea of focusing on a target and using these fragment screens. And I then went to another student, so this is Haran, and this we needed a chemist, so I poured in my collaborator, Chris Abel, into this, and we screened crystals initially with fragments, and we set it up. I had 2 people in my lab, and 1 in Chris's, and then we showed that it worked, and then we got $500,000 for that from another student who was my student in the 70s, who was running a venture capital group that I've been involved in. And he said again, Tom, you know, this won't work, and I'm only giving you $500,000 because at the end of the year you come back with another idea that's better. Now this time I was right, because as you can see, 18 years later, it was actually 14 years later, we sold it for nearly $1 billion. So this time I was right. I'm not always right. But we got 20 people initially, 20 million borrowed. We got 80 employees after a little while. We got a building built for us. We're still in that building, but extending now very fast. And this is the methodology. So what you're doing is you're decreasing complexity, you're increasing promiscuity of the molecule, and there you can see it's bound in there. You get your chemist, your computer person, and your crystallographer around, and you say to the chemist, you know, do you want to elaborate it this direction or that direction? So So, the biologists will say, well, this is more interesting. The chemists will say, but this is chemically more easy. And so you can work together as a little group. We turn this around every day, and you can see we go from 1 millimolar binding, very low affinity, down to low nanomolar. This is on a typical product. This is a kinase project from the company. We can do that in about 30 weeks. We've done it around 30, 40 times in the company. And so what happened is we ended up in 2012 with a whole lot of drugs, more than 10 I think at this time, in clinical trials, some of them going to phase 2, phase 3, everything getting very, very expensive. We had to do something. Now what did I want to do? I didn't want to sell to AstraZeneca, GSK, Pfizer. I knew them too well. They would acid strip us and close us down. They did that to a lot of other people. I needed to find a family firm, and I found it. Well, I didn't find it. They came to us actually, but I identified exactly what I wanted: a family firm with an old guy there, a younger guy involved, and grandchildren hoping to gain from the company. Not a capitalist setup with a 4-year chief executive hoping to make big bonuses. How can you have a bonus for somebody who works for 4 years when it takes 14 years to make a drug? It's not the right incentive. So chief executives do mergers and acquisitions and often destroy the science and structure. So we have a family firm in Otsuka, as you heard, 886 million, but they've left us as we are, and I'm still on the board of the company, which is still called Aztecs, and it's wholly owned instead of all of us sharing. And we got our first milestone with the US FDA filing last November, and then just 2-3 months ago, we got our first approval. So this is for breast cancer. It's the first-line treatment for breast cancer. It's a combination therapy, and it's being pushed through by Novartis that we've collaborated with, and they've been extremely good. So that's a nice story. We're still doing it. We're still up there. I go up regularly, and we're still involved. But what can we do in academia? So 2 things I'm going to have to accelerate here pretty quickly. One was to neglected diseases, and the other is very complex cancer targets in cell regulation. So I'm going to whip through this. So in tuberculosis, of course, we've had only 1 or 2 drugs in the last 30 years, and we're getting resistance coming. And Bill Gates sent his man to our company and wanted Aztecs to be involved in this. This was about 10 years ago. And we learned there's Bill Gates' interest, and this area was particularly interesting to me. Huge tuberculosis in an area where I'd seen my relatives in Zimbabwe dying of AIDS, but of course others getting infectious diseases like tuberculosis. And so what we decided to do was not to develop it in the company, but to spin it back into the university. So Chris Abel and I set up a sort of mirror image of the company, and Aztecs helped us. And this is my family. So this is my wife, who's got a doctorate, and I think she's had a paper in Nature every 5 years for 35 years, and retired this year. These are my 2 daughters about 10 years ago, and these are my grandnephew and niece, and they're the families that are affected. So we've lost 1 person through AIDS, and 2 people I've been trying to rescue, one by getting them imported into UK with tuberculosis. So it's a beautiful country with a terrible crisis. And so what we've been doing is running in academia, and I just wanted to show you a totally multidisciplinary type team. So you heard the talk from Marcio, who was one of the first ones in the academic group, but working with another Portuguese speaker, Vitor Mendes, who's now running it, but also you can see Indians, Polish, And this is a bit untypical, but I assure you we have 50% women in the lab at any time. I'll show you the whole team in a moment. So we go through all the screening, we've set it all up in the lab. It uses all to make sure academic available things like SPR or thermal shift is easy to do. And we test all the hits out. We're doing X-ray, NMR, ITC or surface plasma resonance. And this is a picture of one case where we've got lots of fragments. And you can just see how easy it is to design a molecule when you've got these fragments hitting like this. It's very beautiful. This is Bill Gates. I got a phone call from Seattle saying that Bill wanted to come spend the day with us. But for God's sake, the message came, don't tell anybody in the university, because they're hijacking. So he arrived on his own. This is Chris Abel and myself walking him up the steps in my lab. And everybody in the lab was just asked to introduce themselves by saying which country they came from, which language they spoke first, and which discipline they had, just showing the interdisciplinarity and multidisciplinarity. And Bill then just put on a lab coat, he talked to them, and then he debated. And this is one of our team being asked the question. And Bill was saying, where's the waters? And he's looking at a picture here, just over there, with the drug molecule and no water. So, you know, Bill has been very interested in water structure. I had lots of debates with him. He's invested in Schrödinger and other companies to model waters. And so the reply came back very quickly. He said, oh, we've removed the waters so you can see the drug. That was a very quick answer. He went away feeling happy. So we've been taking that through. We're doing other mycobacterial diseases. This is Mycobacterium abscessus. Again, we've got quite a lot of funding. And then we've also been trying the same things on cancer. And the way I do it in academia is I take a paper like this. This is homologous recombination. We published it in Nature in 2002. And we defined the pockets of the interaction between BRCA2, which carries around 51 into the nucleus to mediate homologous recombination. So I had that structure, and I just took it to Wellcome Trust, and I said, give us some money, and we'll make a novel drug on the protein interface. That was 2006. It took us a long time. So we set up the assays. This is just a picture of what the structure looks like. So this folds on to the globular protein, and you've got two pockets. This is what we call the anchor pocket, and this is an alanine. So that binds first, then this all folds onto the surface. And you can see this residue here and here is absolutely conserved, so you can see how important it is for the function. And what we've done is we set up assays. This is Ashok Venkataram. He's a medic who works up in Addenbrooke's, and he derived an assay. I'm not going to describe how it works, but it involves a lot of cell biology. It's quite straightforward. And we did send this later to some more referees, the referees said it would never work. I keep showing this picture because I have, I think it was 740 crystal structures of it with molecules that bind. So never get upset if the referees say your experiment won't work, because they're often wrong. And they usually look backwards instead of forwards. So we need a good team. So this is Marco Huberman, who comes very, very often to Brazil and has taken over all of this. He led the team the first grant between 2006, but being paid on it. Then he became a professor in the university. This was my last act before I finished being head of department. And he's now taken over all of this drug discovery side. But I just want to make the point that it involves biochemists, crystallographers, chemists, and clinicians to run it. So that's where we are, and just a final thought. What are we going to do about resistance? So Douglas and David have been working on that. David derived an amazing database, because there wasn't really the information for machine learning, and it's very critical. So I think that contribution is as important as the software idea. You've got to have reliable data for machine learning, and that we can then turn into a system that predicts the effects of mutations on ligand binding. So we now have software to predict resistance. And some of the mutations are very close to the ligand, but others are quite distant. And like the other things I was telling you about genetic disease, mutations often occur in interfaces a long way away. And that's because the bug, it wants to get over, it wants to be resistant to the drug, but it doesn't want to be dead. So somehow it's got to get rid of the drug without actually killing the activity of the target. And so the reaction, the responses in resistance are really quite complex. So we've done a lot of analysis on that. And what I'm hoping to do over the next 5 years, if people will allow me good health, is to really look at this question of fighting resistance, repurposing old drugs, and then we've got lots of ideas about fragmentation and regrowing to get them. So I leave you there with the science. I just show 2 more pictures. So this is my team. At a party on one occasion a few years ago, we had a conversation where each person spoke to the next one, but then used a different language than was used before. We found we could do 37 languages in my team. I can only do 2 or 3 and only one very well. So that was all very nice. And then the other nice thing that happened to me last year was to get elected to your academy, as you've heard. And so this is, I think this was in Rio last year, wasn't it? And you can see your distinguished members of the academy here, and I feel absolutely excited and delighted, as I do when I'm always in Brazil, but that like this is a very special occasion. And there's me standing, showing my certificate last year. So I leave you with those thoughts, and I hope you can understand why I've been really fighting the university, trying to make me retire. I've now beaten them off for 8 years, and they've given me another 5 years. Science is just fantastic, and if you can have lots of friends around the world, you can interact, and you can have multidisciplinary groups. What else would I want to do? And I hope you all feel the same about your science and the way that we all interact. Thank you very much. Thank you very much. I hope they got a drink for us all.
Well, my dear friends, ladies and gentlemen, this ceremony is closed. Thank you very much for your presence here. Have a pleasant night. Bye.